All righty, we are back from break. And we have arrived at the Innovators Forum, an exciting session about companies that are innovating in HD. Love this session. It's right now. It's happening right now. So to give an overview of current strategies for HD therapeutics, please welcome Dr. Sarah Hernandez from the Hereditary Disease Foundation to kick off this session. Come on up.
All right, sorry guys, I'm gonna try to put my computer here. Hi everyone, I'm Sarah Hernandez from the Hereditary Disease Foundation. Sorry, AV crew, I'm gonna totally manhandle your mics here. All right, so I'm very excited to be kicking off this session because at the HDF, I get to deeply focus on HD research, and so it's a very great honor to share with all of you some of the current research strategies in HD. All right, so treatment strategies really fall into two categories. Those that are designed to treat the symptoms, so essentially to improve the quality of life of people living with HD, and those that are aimed to modify the disease course. The disease-modifying treatment that's really furthest in the clinic right now overall as an approach is Huntingtin lowering. And people are taking a variety of different strategies in the way that they're going about this. And so people are using antisense oligonucleotides, they're using splice modulators, they're using gene therapy, they're using RNAi, and really all of those can fall into two flavors, really total Huntingtin lowering or allele specific, and I'll talk more about that in a minute. For gene therapies, people are either taking viral approaches where they're using things like AAVs, or they're using things like LNPs, lipid nanoparticles. And really LNPs can be used for any one of those, but for the purpose of this talk, because people are using it for gene therapy, I'll focus on that area. And then there's also other disease-modifying approaches that people are exploring. So things designed to target somatic expansion, which people are using splice modulators for, and then also cell replacement. And so for the rest of my talk, I'll kind of hit on each one of these. I'll fill in certain details and talk about different companies that are working in these arenas, and then I'll come back and fill in some of those details. All right. So first I'll touch on symptomatic treatments, and really these are aimed at improving the quality of life of people living with HD, really designed to treat things like depression, to treat chorea, and also medications aimed at treating cognition. And so a major disclaimer here, I am not a medical doctor, so I am not going to go into all of the medications here or the indications for which they should be used, but I do just want to highlight that there are approved medications that can be used to treat the symptoms of HD. And so for things like depression and psychosis, there are antipsychotics and antidepressants, which people can work with their neurologist to determine if that would be the best use. For chorea, there are regulatory agency-approved medications from Neurocrine Teva, so we have things like Ingrezza and Austedo, and then also Som Biotech is working on a drug for chorea. And for cognition, Sage Therapeutics is advancing their drug called dalzanemdor, previously known as Sage 718, that they are hoping will help with thinking and memory in Huntington's. All right, for Huntingtin lowering, really as an overall approach, this is what's furthest in the clinic. This is what the most people have tried. This is what we know the most about from the most number of companies. And so there are many companies exploring this in various ways, but essentially their goal is the same. They want to lower the Huntingtin message, which will lower the Huntingtin protein. And so people are taking two approaches to this. They're either targeting total Huntingtin, so they're getting both copies of the Huntingtin message from mom and dad, or they're specifically going after the expanded copy, so they're using an allele selective approach. And so the first Huntingtin lowering out of the gate was antisense oligonucleotides, or ASOs. And essentially this is a short fragment of genetic material, shown in red, that's complementary to your gene of interest, such as Huntingtin, shown in blue, forms a double-stranded RNA molecule, which your cells don't like. It degrades it. It doesn't bother making that protein. The first company out of the gate for this was Roche and Genentech, and so they were testing their total Huntingtin lowering ASO called tominersen, still being tested in a phase 2 trial. Wave Life Sciences was almost immediately behind them, testing their allele selective ASO, and they're on their third iteration of that ASO now, called Wave 003. They're testing that in the clinic. And lastly, I'll mention here Vico Therapeutics. So they're also testing an allele-specific ASO, but what's unique about their approach is they're specifically targeting the CAG repeat. And so in theory, if this works for something like Huntington's, it could also work for other CAG repeat disorders. And so actually they're testing this right now in a trial with HD, as well as patients with spinocerebellar ataxias 1 and 3 in a basket trial. And so in all of these trials, ASOs are delivered via lumbar puncture, but the advantage of this approach is that these are designed specifically to target Huntington, and so they should be specific for that target. And I could envision things like this being given every few months. The interval is being tested in several of these clinical trials. Another approach that uses a genetic component is RNA interference, or RNAi. And in this approach, a synthetic piece of RNA is delivered to the cells, and it takes advantage of the cell's own machinery through the RISC complex to produce a genetic fragment which lowers the gene. And so there are two companies right now taking this approach. There's Atalanta Therapeutics and Alnylam Pharmaceuticals. They are both speaking in this session, so I will let them tell you more about their own treatments. Except I will say, and not to steal the thunder too much from you, Alnylam, but just this week we learned that Alnylam is recruiting for a phase 1 trial, and so that's incredibly exciting. I hope they tell us more. All right, and both of these, for Alnylam anyway, it will be delivered via lumbar puncture. For gene therapies, people are trying currently viral delivery, so things like adeno-associated viruses, or AAVs. And so in this approach, there is genetic material encapsulated in a virus, like this purple blob, that gets fused into the cell. It delivers the genetic components, and it gets replicated by the cell. So it's important to note this is non-integrating, so this is not going to change the host, the genetic host, the genome of the host. And currently, Unicure is the furthest in the clinic with this right now. So they're testing their drug, AMT-130, and so this is delivered via brain surgery through 6 separate injections. A company called Lattice Bio, they recently received a $1 million award. They were part of a team that received a $1 million award from the Hereditary Disease Foundation to advance their AAV-based therapy. And so what is advantageous about their approach is that it's incredibly potent. And so this would just require 1 injection within the brain at a single drop of liquid. And so they're currently testing that in non-human primates. And lastly here, I'll mention Spark Therapeutics and LifeEdit. They're both advancing AAV-based treatments. They are also both speaking in the session, so I'll leave it to them to tell you more about that. But currently, AAV-based approaches will be delivered via brain surgery, which is a major drawback of this approach, obviously. But the major advantage is that all of these are one-and-done treatments. So in theory, people would just need this treatment once, and Huntington would be lowered for life. Another method for delivering gene therapies is through lipid nanoparticles, or LNPs. And so these are little bubbles that are made up of lipids that can carry lots of different types of cargo. So not just genetic components, but also things like CRISPR. And so this is a major advantage. And Incisive Genetics is a company that's exploring this route, as is LifeEdit. And so LifeEdit is speaking in this session. Hopefully they'll tell us both about their AAVs and LNPs. But a major advantage of the approach for LNPs is that they're really diverse. So not just in the cargo that they can hold, but also in their delivery method. And so right now, these types of approaches would require brain surgery, but really you could deliver them in almost any way. So think of your COVID LNP-based vaccines that you get straight into your muscle. And so they're really powerful. Another advantage that they have over something like an AAV is that LNPs can be produced at much larger quantities for a lot less money. And so it's a major advantage of working with LNPs. Another method that people are using is something called splice modulators. These are small molecules that essentially work to splice the Huntington message. It moves up the stop codon. The cell sees it doesn't make any sense, and it doesn't bother producing it. And so as an aside, I would say if there's any aficionados with creating figures in the audience that are so inclined, I would strongly suggest a splice modulator figure for Huntington's, because I spent an embarrassing amount of time Googling for one, and that is why I present you here with a stop sign in a random field, because that's the best I had. All right, okay, but I digress. So PTC Therapeutics is a company testing their small molecule splice modulator called PTC 518. We recently had good news over the summer that it seems to be safe and well tolerated, so that is advancing through the clinic. Skyhawk Therapeutics has a very similar molecule called Sky 0515 that is also being tested in the clinic. And so these small molecule splice modulators, they're exciting because they could be taken as a pill. So this is something you could envision as taken once or twice a day. It's very non-invasive. But one of the drawbacks is they're not specific for Huntington. And so a lot of energy and effort is put into understanding the off-target effects that these drugs may have. But perhaps serendipitously, it seems that one of the off-target effects might also allow them to target genes associated with somatic expansion. And it seems almost too good to be true that these would allow for that kind of 1-2 punch against Huntington. You're lowering Huntington and maybe targeting somatic expansion. More data is definitely needed, but it's a really exciting area. And so for those who may not know, if you weren't in the last session with Vanessa and Ricardo, somatic expansion is this perpetual expansion of the CAG repeat that we see over time. And so this is a figure I really like from a preprint by Steve McCarroll's group that I think describes this really well. And so on the bottom, you're seeing all these dots, these little gray dots that represent neurons that are vulnerable in HD. And so when someone's born with the pathogenic length, the cells spend most of their time under that blue curve. But eventually, over decades, they start to expand up to about 100. And then they expand more quickly. In a matter of years, they can get up to 150. And then they expand even more quickly, where in a matter of months, they can reach up to 450 CAG repeats or higher. And so it's really this accelerated CAG repeat expansion that cells are gaining that scientists think could be leading to the toxicity that's experienced in some of these vulnerable cells. And so this is something that's unique to vulnerable cells, specifically brain cells. And so the thought is, if we can control this, if we can get a hold on this, we could keep these cells healthier, we could perhaps slow signs and symptoms of HD. And so this is why it's so exciting that perhaps those Huntington-lowering small molecule splice modulators control this, because they're not only going after the nefarious Huntington protein, but they may also be starting to regulate somatic expansion. So it's incredibly exciting. But there are also companies that are specifically working on somatic expansion. There's Locus 23. They have a small molecule that targets MSH3 within the somatic expansion pathway. We recently found out they received $43 million in funding to advance this program, so hopefully we will hear more from them soon. And then Argenta, who are advancing a small molecule that targets PMS1 within this pathway. Argenta are speaking in this session, so I won't say more than that. I'll just say that they can be taken as a pill, which is a really exciting format. Another approach is cell replacement. And so people could do this either by adding back stem cells to the brain and turning those into neurons, or they could convert cells that are already in the brain that don't die in HD into neurons. And so there's a company called Nuexcel Therapeutics that has a cocktail of small molecules that can be added to the brain to convert astrocytes, which don't die in HD and replicate, and turn those into neurons. There's also an academic lab run by Don Cleveland out of University of California, San Diego, taking a similar approach, converting astrocytes to neurons. And then another approach that people could use is they could add back stem cells to the brain and turn those into neurons. And so Sana Biotech is taking this approach. They are speaking in this session, so I won't tell you more about that. But Leslie Thompson, separately at the University of California, Irvine, is also doing this. So she's adding neural stem cells to the brain and turning those into neurons. And she recently shared with people at the recent HDF conference that her lab is advancing this toward trials. They're gearing up to start a phase 1-2 trial with this approach, which is very exciting. And so currently these would be delivered via brain surgery. Lastly, the last approach I'll mention that doesn't really fit into one of these boxes that I've described is targeting the sigma-1 receptor. And so the sigma-1 receptor is on the endoplasmic reticulum, and it aids in communication with the mitochondria. It's thought that if we can target the sigma-1 receptor, it would help neurons live under conditions of stress. And so HD causes all sorts of effects. If we were to activate the sigma-1 receptor, it could alleviate some of those symptoms and hope slow signs and symptoms of HD. And so Prolenia are advancing a sigma-1 activator, which can be taken as a pill that they've tested in several clinical trials. All right, so we've covered a lot of ground in just 15 minutes. And so what I hope you can appreciate really is that there's a lot of different people that are working in the space. They're working on a lot of different strategies to get these treatments to the clinic. And this has become one of my absolute favorite slides to show in conferences. It was originally created by Leora Fox, so I'll give her a shout out for this. And over the years, we've all added to it. But what you're seeing now are names of companies that have either had a presence at an HD conference, they've made a public commitment to working on HD, or they have an ongoing HD clinical trial. And so what I hope you can appreciate is there's a lot of people working on HD. They're all taking different approaches, and they really will develop a treatment that will get us to a disease-modifying drug. And so I appreciate everyone listening to this overview. I'm excited to sit with you in the audience and hear more from some of these companies on strategies for HD therapeutics. Thank you.
Great job. Thank you, Dr. Hernandez. Great job. So before I bring up Dr. Ross, I understand there's some questions about what's on the table, the little candies. So real quick, I just want to explain. We can thank Dr. Kostick for them. These are her creation in many ways. They are called Buckeyes. They are native to Ohio. They are deliciousness in a little candy wrapper. It is a peanut butter ball with chocolate. They're dipped in chocolate. Unless you have a peanut allergy, they're not for you. Please don't eat them. And I don't know the rest of the history, but you can ask Dr. Kostick. She's happy to explain it to you. And I know they're just native to Ohio, so they were just a little treat for you to give you a little sugar boost while we continue, okay? Right? Have you had them? They're good, right? They're addictive. All right. So now we can welcome you, Dr. Ross, Chief Scientific Officer of the HSG, to moderate the rest of this session. Come on up here, Dr. Ross. Thank you.
Thank you very much. And thank you, Sarah. That was really such a great introduction and overview of all the different therapeutic approaches that are being tried. And I think one of the reasons why, after a couple of setbacks in the past few years, we're extremely optimistic again about all the different possibilities. So this is going to be the Innovators Forum. This is traditionally our session in which companies or programs that are not yet in the clinic or just starting out in the clinic present their work and their ideas, and it's often a showcase for novel approaches to therapeutics, and many of the programs that have been presented in this forum then later end up in the clinical trial roundup and in clinical trials. So without further ado, I want to get started. I just want to mention about timing. So we have 75 minutes and 6 talks. I'm not going to give long introductions, so if people can get up and down quickly, we have 12 minutes per talk. And what I would like to see is 8 to 9 minutes of presentation, and then that'll give us 3 to 4 minutes for questions afterward. And our first talk is by Kevin Sloan, who's Vice President for Development Programs at Alnylam Pharmaceuticals.
Thanks, Chris. Those Buckeye balls are fantastic. I've had too many of them today, and I've had like this ongoing sugar high. Great. So, quick disclosures. I'm an employee and shareholder at Alnylam Pharmaceuticals. We all know Huntington's disease is progressive and fatal, and that it's driven by the mutant Huntington gene, or HTT. The mutant gene is toxic, conferring a broadly disruptive gain of function, driven by CG repeat expansion and the underlying somatic instability. There's hallmark protein aggregation and widespread neurodegeneration. Now, there's a rapidly evolving body of preclinical research suggesting that both the full-length mutant Huntington, but also the shorter exon 1 splice isoform form of mutant Huntington likely contribute to disease pathology. So we believe that investigative Huntington-lowering approaches may offer a potential to alter the course of disease progression, but the safety and extent of achievable clinical benefit have yet to be established, and some early efforts were limited by therapeutic platform challenges. We're still learning how to do this. What's the right hammer for the nail? So RNA interference may be new to some of you, but it's actually fairly archaic. It's an endogenous biological pathway that exists in all of us. It was first described in plants in 1990 and then characterized in a variety of animal species for the next decade or so. Alnylam was founded in 2002 with the vision to try to create a new class of therapeutic medicines to harness this endogenous pathway. And we spent about 20 years at this point really trying to optimize synthetic RNAi drugs based on this principle by engineering in safety and potency and durability and specificity to really make these work. And that's given rise to about 5 approved medicines at this point, all of which go after targets in the liver. And then more recently, in around 2018, we learned how to get these into the central nervous system. Delivery is always the challenge with these technologies. And for the last couple of years, we've been going after a number of really interesting genetic diseases in neurology. So RNA interference is a natural biological process to regulate gene expression. It happens in all of us. We can design and engineer a synthetic gene-specific siRNA in the top left. That's our drug. And we can introduce that into the cell where it loads into an endogenous protein complex called RISC, the RNA interference sciencing complex. Once it loads into RISC, all we're doing is creating a zip code. We're programming that complex where to go to seek out and destroy a specific target messenger RNA. And by destroying that, we reduce downstream protein expression and turn off the specific protein. Now there's a catalytic mechanism, so once the siRNA molecule is inside, it can load and reload that RISC complex, and so a little bit of drug goes a long way. And what we've learned from our work is that RNA therapeutics typically have a very potent and durable efficacy, supporting infrequent dosing regimens in clinic with good safety profiles. And importantly, RNA therapeutics are a unique class of genetic medicine. They're quite distinct from antisense oligonucleotides. It's a different molecule, it's a different biological pathway. What's similar is the end result, reduced protein expression. So over the last few years, we've achieved the first human translation of RNA interference with this platform in the central nervous system. And we did this with a molecule targeting amyloid precursor protein, or APP. This has therapeutic applications in Alzheimer's disease, cerebral amyloid angiopathy, and other indications. And we learned first in our non-human primate and preclinical work is that after an intrathecal dose of our molecule, we would get very widespread distribution and target engagement throughout different regions of the brain. On the right, there's data from a clinical study. This was a phase 1 single ascending dose study with this molecule. The y-axis shows APP protein in the CSF. The x-axis shows time. And what you see is after a single intrathecal dose, rapid lowering that persists for 6 months and then recovers for the next 6 months. So that graph is showing you 12 months, 1 year after a single intrathecal dose, sustained target engagement, sustained lowering of APP. So we're really encouraged with the durability and potency that we've seen as we've moved this platform into the clinic. And more importantly, we're very encouraged by the safety profile. Because of the history of oligonucleotides in the CNS, we paid a lot of attention to CSF safety labs, looking at neurofilament, all sorts of parameters, and this platform appears very, very safe. So this molecule is now moved into phase 2 testing for efficacy, and we've taken this now clinically validated delivery platform and applied it to a collection of other targets in neurology. So ALN-HTT02 is an investigational RNAi therapeutic designed to reduce Huntington protein expression in the CNS. And if you think about a typical Huntington's patient, they have 2 copies of the gene. The wild-type allele on the left, with the first exon, a normal stretch of CAG repeats, and then quite a lot of downstream gene after that. On the right is the mutant, the same first exon, an expanded CAG repeat tract, which wreaks havoc, causes the problems, and then the bulk of the downstream gene. And we took quite a bit of extra time to engineer ALN-HTT02 to recognize a conserved sequence that lies within that first exon. It was a challenge. It's a short, restricted gene space, but we were successful and found a molecule that can do this. And why this is important is the molecule is designed to reduce expression of all Huntington protein species. So we will turn off and reduce the full-length mutant Huntington protein that we've typically talked about in meetings like this. But we'll also be able to turn off the shorter exon 1 fragment mutant Huntington protein, which contains just the exon 1 and various expanded CAG repeat lengths, but not the rest of the gene. And if you're following the recent academic literature, these shorter fragments, there's a lot of evidence in mice, in flies now, that shorter fragment alone can drive disease pathology. So the thinking here is one probably has to engage both of these species to have effects. Now, because that target sequence is so heavily conserved, it also exists in the wild type allele, and so we will also turn down expression of wild type Huntington. So I'll show you a couple pieces of data, and because of the debate in this field about the safety of Huntington lowering, we've done a lot of work in rodents and non-human primates to try to characterize our molecule, the safety of not only the molecule, but also the safety of Huntington lowering. So this experiment is just a single dose of our candidate in a non-human primate. And the way these two graphs are going to be set up is huntingtin protein remaining on the y-axis, and in this case, 2 different time points, 3 months or 6 months down on the x. So after a single dose, the first group of bars is 3 months. Each pair is a low and a medium dose level, and then they just go in order. Cortex, hippocampus, caudate, and putamen. You see widespread lowering and target engagement throughout the brain regions. At 3 months, go out to 6 months, we start to see some recovery, which is fantastic. We don't want a forever drug. We want to be able to control it through repeated administration. But that durability profile is very similar to what we had seen for APP in our primate studies before we went to the human clinical work. So we're really encouraged by the durability. We anticipate this will support infrequent dosing in clinic, maybe every 6 months, maybe every 12 months. We'll learn that as we go through clinical studies. The safety profile here was very encouraging through 6 months. We had no in-life neurological abnormalities and no elevations in CSF, NFL, or total protein. We've gone on to push the dose and push the exposure and push the huntingtin lowering. So this is now another non-human primate study. This time we're looking at multiple doses over 6 months, and we're pushing the dose really as high as we can, not because of safety, but because of just how we can formulate the molecule itself. And so here you can see huntingtin protein remaining on the y-axis, and then just stick to the right side for simplicity, CSF control in the gray bar. The light blue is the same low, the medium blue is the same mid, and now there's a higher dose, the max feasible dose we can administer. And what you can see is we see very deep huntingtin lowering in these animals in excess of 90% over 6 months in the cortex of primates. And we're really trying to push it. We were looking to see if we could expose some safety issues in these primate studies, but what we saw was really nothing. Very encouraging safety profile, no in-life neurological abnormalities, no adverse CSF parameter changes, and no adverse microscopic findings after a very thorough histopathology workup as part of a GLP tox package. So this molecule has been evaluated in 4 independent NHP studies to this point. Across all of them, we've seen no adverse findings, even after very deep huntingtin lowering beyond 90%. If you're interested in learning more, my colleague Will Cantley has a poster at this meeting where we're trying to share with the community a lot of our learnings from these primate studies beyond these. I think it's number 25. I encourage you to stop by at the poster session. So with the profile of this molecule engaging exon 1, the safety we've demonstrated, the potency, anticipated durability, we have gone on and now initiated a phase 1b study of ALN-HTT02 in adult patients with Huntington's disease. This is a placebo-controlled single ascending dose study to evaluate safety, tolerability, and PK/PD. The study population is age 25 to 70 years with more than 39 CAG repeats, HD-ISS stage 2 and early stage 3. Importantly, anyone who comes into the study and is randomized to placebo in the first part will have the opportunity to receive a dose of active study drug in the second part. So everyone who joins the study to participate will have access to study drug. It's initiating in the UK and Canada. We're hoping to add a couple additional countries very soon. The QR code at the bottom will take you to our posting at ClinTrials, or you can just search ALN-HTT02 with a dash. So to summarize, Alnylam's clinically validated siRNA delivery platform may offer a new approach for huntingtin lowering in the CNS. ALN-HTT02 is an investigational RNAi therapeutic designed to durably lower all forms of mutant huntingtin, including the exon 1 fragments. And we believe there's potential to optimize the depth and duration of huntingtin lowering through clinical dosing regimens to really explore how to get the most efficacy while still preserving safety with this kind of approach. Now, wild-type huntingtin lowering in the CNS appears very well tolerated in non-human primates after intrathecal dosing with this molecule, and those findings have been demonstrated in multiple independent studies, even with very deep huntingtin lowering. And finally, a phase 1b study of ALN-HTT02 in people with Huntington's disease is currently initiating. With that, I'll say thank you. Really appreciate the collaboration and the support this community has given us over the past couple of years, and we look forward to sharing more progress as we have it.
Excellent. Thank you very much. We have a couple of minutes for questions. Maybe I'll start out by asking, how does the distribution of, is there a microphone that people can come up to, or do I see you're getting a microphone? Great. How does the distribution within the brain of the siRNA compared to an ASO in terms of when you do intrathecal injection? Does it get as far as the striatum as well as an ASO, less well than an ASO, better than an ASO? Just on kind of a broad
Yeah, I mean, I think the challenge in the human, there's no data to point to, right? I mean, the human data with the ASOs is due to the small number of post-mortem samples, I think Blair Levitt and others have been involved in looking at. And I think unless there's some really unfortunate death in an RNAi trial, it's hard to get that data. I don't think we've done any real head-to-head work in primates. I will say at a macro level, it should be fairly similar, right? The oligonucleotide in general is going to distribute broadly. And I think we've shown the target engagement on caudate and Shaman, the deep brain, right?
Great, thank you.
Yeah.
Question over there?
Yeah, actually, it's a follow-up on your question, Chris. You may not be able to comment on this, but the C16 modification, it really does seem to improve that biodistribution. Do you have can you tell us anything about proposed mechanisms of how intrathecal delivery of a C16 siRNA gives you so much better distribution and how that?
Yeah, I mean, there's a couple of parts to that story, right? And so one of the reasons it took so long to get siRNAs into the CNS and the sort of breakthrough in that sort of 2018 period was if you just inject like an unmodified or non-conjugated siRNA, it gets cleared out of the CNS very quickly and gets effluxed before it really has a chance to have activity. And so, there's this class of like lipid conjugates like C16, when you put it on, it seems to increase the retention time inside the CSF. And so giving it more time allows it more time to distribute through the CSF and then be taken up. In terms of how it's actually internalized into the cell, so our whole historical platform in the liver is based on using a GalNAc conjugate, which binds to a specific ASPGR receptor and is internalized into hepatocytes, a very direct uptake mechanism. So the C16 conjugate may also be taken up through like a lipid scavenger mechanism, but we're still trying to map exactly how that's working. So.
Great, thank you very much.
All right.
If we can get in one of them if it's quick and we get a quick answer.
Quick answer.
Is there any insight to the delay of starting up in the United States?
Any insight into the delay? Slow review times by the FDA. We did announce last week that we have the green light to proceed in the US, so not making a huge deal, but we're here meeting with PIs. Several of our PIs are here at the meeting, and we're trying to figure out how to activate sites as quickly as possible. So
Okay.
watch this space.
Thank you. Shows the advantage of keeping the talk short so that we can get into questions. I have light shining in my eyes, so I can't necessarily see where the mics are, so really wave your hands if you wouldn't, please. Next is Travis Wager, co-founder and president and CSO of Argenta Therapeutics.
All right, thanks for the introduction. And it is a great pleasure to be here. A lot of the kind of road show that we've been doing really is trying to get input from individuals because we are certainly new to the Huntington field. So at Argenta, we're really interested in developing small molecules for historically undruggable targets and really thinking about it through a different lens rather than going after the protein, really focusing on the RNA, specifically with oral small molecules. And so obviously there are a lot of advantages to oral small molecules. One of the biggest is convenience. Another one, obviously, is really getting to the target of interest. So Huntington disease is one of these repeat expansion diseases that we've heard about today. A lot of work has been done by numerous people in this audience, really, developing and understanding, repeats and really the control of those repeat expansion diseases. Obviously, it's sort of that at the DNA level, which I think is important because it really is at that fundamental starting point of Huntington disease, and as these repeats grow. So it sort of falls in this kind of interesting environment in that, while it is a rare disease, it's actually a large rare disease. It's not quite a large disease itself. And so where does it actually fit in terms of people's portfolios? So we're really interested, obviously, in repeat expansion diseases, neuroscience, PMS1 specifically. Vanessa mentioned earlier, a lot of the nice GWAS work that has been done since 2015, really identifying several genes in the mismatch repair pathway. The two that really, moved to the top in terms of a drug development standpoint are MSH3 and PMS1. We like both of those targets. PMS1 is clearly the front runner in our portfolio. And part of that reason is the additional human genetics that go along with it, as well as the safety. There's some nice SNP work or variants that exist within PMS1. One, that actually confers a earlier onset, more severe, phenotype, and that is essentially where PMS1 itself is upregulated, or you see an increase in protein. McAllister Group has done some really nice work last year identifying late onset patients. And really PMS1 is a gene in there that has a very significant, different SNPs or different variants that have either non-synonymous or damaged or loss of function associated with them. And so you have this very nice bookend of upregulation of PMS1, early onset. Downregulation of PMS1 in humans, again, this is all human data, that confers later onset, again, making it a pretty cool target for us to go after. Within our platform, we really have built this very large platform. We've scanned through the entire human genome, looked at roughly 20,000 genes. Of those genes, we can effectively target at the RNA level about 6,000 of them. One of the cool things in here is PMS1 and MSH3 both have actionable target sites. PMS1 has 4. This becomes really important when you think about selectivity, because not all target sites are equivalent. And we certainly heard earlier on where some of these splice modulators that are out there are non-selective or less selective, branaplam being one of them. And then earlier cases where potentially HTT molecules may also have a crossover into some of these repeat expansion targets like PMS1. So the more targets that you bring in, the more liability you bring in from an overall safety perspective. So it is really critical to pick the right target sites. And we've developed this over really the last 4 years that we've been working on this problem. So very similar to what we saw earlier from PTC, our approach really is to leverage all that genomic data to identify these target sites. Normal RNA processing is across the top, where you have that pre-mRNA, essentially the intronic region is spliced out. You make this canonical or this mRNA that goes on to protein synthesis. In the case of PMS1, that's that pathological protein that really drives these repeat expansions. Our approach is to insert this segment into it, of which we've identified, leveraging the spliceosome complex and a small molecule. And you really need those pieces, that small molecule, to essentially insert what I would call a poison exon. And when you insert that exon, it does have stop codons in it. And so effectively, when it gets read, it really goes down this pathway of nonsense-mediated decay. So if there's no RNA, there's no protein. And so that pathological protein does not get synthesized. We basically have worked on this for a number of years now, ran an HTS, have identified small molecules that meet a lot of our criteria around potency, ADME, as well as characterization for what is our human dose. Here's just a snapshot of some of those properties. The nice thing is that RNA and protein track really closely. This particular compound is around 20 nanomolar at the RNA level, and changing that splicing, and protein around 15. So we know they track very well. The RNA explanation is kind of on the bottom, where we have that inclusion transcript goes up at the expense of that canonical transcript, that transcript that drives protein synthesis. That's the one we want to get rid of. And so the beautiful thing is, we can dial this in. It is dose responsive. To the right is the protein. And we've looked at protein, against, kind of different high throughput assays. And that's sort of that top dose response of which we can run hundreds of times. to a slower Western blot analysis, looking at multiple tissues. This is a really kind of a summary of a lot of work that Abraham has been doing. He's got a nice poster on the outside. I encourage you to talk to him because he has basically put his entire 3 years into this project as long as he's been at Argenta, and over that time has really, progressed this program forward. I would like to kind of just stop and thank people out there who have helped Abraham, because there's decades of research that's gone into creating a lot of these models. And if we had to do that on our own, we wouldn't be where we are. And so that's one of the nice things about the Huntington community, is it really is a sharing community. And I think Abraham can attest to that, because some of the data that he's generated is just really amazing. This is just one experiment of many. In here we've run experiments that are 80 days or longer. This particular experiment's 30 days, but still a very long time, where we look at RNA, we look at protein, and we also look at repeat expansions. And across the board, we see robust dose response. And so the RNA, at the end of the experiment is looked at, effectively a nice dose response. As the dose or exposure of drug increases, the RNA drops. The same is true with protein. That's the next slide over with sort of the Western blot. As the dose increases, protein is knocked out. Okay. And when we look at, effectively the CAG size, you have in here your vehicle group, which grows substantially over the course of this 30 days. At a 50% knockdown of RNA and protein, you have a 70% stalling of those repeats. And 90% completely stalls it. And this is quantified in sort of that fragment analysis where you look at instability index as well. A couple more slides. Really, drug distribution, this is one of the things that always pops up. Are you getting to the target? In this slide, we effectively have PK/PD. So the first is, as our drug exposure increases, we knock down the overall transcript and protein. When we look at monkey, right, so this is all monkey data, where we're going in and specifically looking at different brain regions, different tissues, both peripheral and central. We see robust overall exposure. And then when you go into brain and you look at these different compartments, including striatal, we can show a robust knockdown of protein and RNA, very similar to that of what we see in cells. We know it's durable, so we've run studies out to essentially 28 days or longer. We're able to knock down transcript, we're not able to knock down protein at incredibly well-tolerated doses. So again, really showing that we can hit our target, we have that target engagement, which will translate to a robust reduction of repeat expansions. 1 minute. Yep. I'm not going to talk too much about this slide, but really more just to get the audience and people to come up to Argenta and help us think about our clinical design. So obviously, when we get into the clinic, we want to make sure that we select the right patients. That we learn from everything that you've learned from running those clinical experiments, because that's what they are. Let's leverage that information to really come out on the other side with a positive, early sign of efficacy experiments. And with that, I really just need to thank the Argenta team, who really have dedicated their time to trying to solve this problem, to find a cure for Huntington's disease with an oral small molecule. And then we have a ton of scientific advisors. Many of them are in the room. We wouldn't be where we are without you, so thanks for that as well. And with that, I'm going to stop. 1 minute.
Okay, thank you. We're pretty much out of time, so let me just ask one quick question. So these modifiers will alter the rate of expansion and therefore change the onset. Do you have any sense of what the magnitude of the effect would be if you had even, say, complete inhibition of PMS1?
So, well, definitely if we have complete inhibition, of PMS1, we stall those repeats. And so if you go back to, a lot of the work from Steve, the sooner you can intervene, the better off you're going to be, right? Because you get to sort of that potential cutoff of 150, that size is where things really go haywire. If you can get in there before that, you never hit that number. And, people should be basically steady.
Right. Thank you very much. Is this on? Yeah. So to leave time for questions and discussion, I would just suggest that people not give any introduction about Huntington's disease or most of these mechanisms, since Sarah went over them so nicely. So let's see, our next talk is Logan Brown, Senior Scientist at LifeEdit Therapeutics.
So thanks to the organizers for the opportunity to present some of our work in Huntington's disease. Just started my disclosures. I am an employee of LifeEdit Therapeutics and a stockholder in Elevate Bio Company. And so just a really brief introduction to LifeEdit Therapeutics. We were founded in 2017 based on a collection of proprietary CRISPR enzymes. So thanks to Ricardo for giving a really excellent primer on CRISPR approaches to Huntington's disease. But just in case you missed it, there's sort of three main components of a CRISPR system. There's the nuclease that cleaves the DNA. At LifeEdit, we call these our LifeEdit genes, or our legs for short. There's the guide RNA, which is a programmable RNA sequence that determines the targeting specificity of the nuclease. And then there's a PAM, or a protospacer adjacent motif, which is a really short recognition sequence that determines where a cut is going to be made in the genome. And so each one of these nucleases has a unique PAM sequence that it recognizes. And so by having a collection of nucleases with different PAMs, it really allows you to go after lots of different genetic sequences that are linked to the disease genes. So our systems are also very small. You can see in this table the comparison of some of our lead nucleases compared to SP-Cas9, which is sort of the most widely utilized CRISPR-Cas system out there. And this allows us to package everything into a single AAV vector for an all-in-one delivery approach. And so we are developing LEDI-101 for Huntington's disease, and so we're going to package one of our CRISPR enzymes and guide RNA and all of the regulatory elements that are required into a single AAV5 vector and then deliver it via direct intrastriatal administration to target the medium spiny neurons. LEDI-101 is selective for the mutant Huntington's allele based on the PAM-forming SNP 362331, which is in exon 50. And so when this SNP is a thymine, it generates a PAM for our LEGB nuclease, but it does not when it is a cytosine. And so our target patient population are going to be those individuals that have the SNP in exon 50 as a thymine identity, and that needs to be on the same allele as the CAG repeat expansion in exon 1 of the gene. And we estimate that this will capture about 30% of the total HD population. And so just to emphasize that, because we are editing the mutant Huntington's gene itself, this should be a one-time treatment with a lasting therapeutic effect. So it doesn't rely on continued transgene expression from an AAV episome following administration. So the first thing we did was test the ability of our system to selectively edit the mutant allele. So we got a collection of patient-derived fibroblasts, all with the appropriate genotype that we would be treating in the clinic. And then we delivered with either an mRNA encoding our nuclease or the active guide RNA or one of several different control guide RNAs or nuclease by itself. We then harvest genomic DNA and assessed indels or insertion deletions by next generation sequencing. This is a measure of editing on target. And then we also looked at changes in wild type and mutant protein by Jess capillary electrophoresis or by traditional Western blot. And so in the left panel here, we're just showing that we've only observed editing of our target mutant allele when it contains the T-SNP. We never see editing of the C-SNP containing allele with the wild type gene. And we only see editing when we have our active guide RNA, none of our control conditions essentially. And then similarly, if we look at the right panel, we've only seen reduction of mutant protein in our patient-derived fibroblast lines or a healthy donor line that did not have the target SNP. And we did this by capillary electrophoresis, but we've also confirmed this by traditional Western blot in all of our cell lines. And so for off-target analysis, we start by identifying all possible off-target sites using a three-rung approach. The first thing we do is an in silico prediction based on homology to the guide RNA sequence. Then we do a population variance analysis to identify potential genetic variant sequences that could introduce unforeseen SNPs or unforeseen off-target sites. And then we also use an unbiased biochemical method, digenome-seq, in which the patient DNA is digested with our enzyme for an extended period of time. And so these three methods altogether give us a list of 300 putative off-target sites. We then edit patient cells again, as we did previously, and then we do amplicon-based next-generation sequencing, again, looking at all of those putative off-target sites. And we have not seen any bona fide off-target editing to date. We do have some additional experiments to follow up with these, such as using optical genome mapping to look at chromosomal translocations. So for PK/PD modeling, we've been using the BAC-HD transgenic model. These guys carry a full-length human mutant Huntington's transgene. Really importantly for us, it has the exon 50 target T-SNP, which is required for our targeting system, and they express the full-length mutant protein. And so at the top here, you can see our AAV construct schematic. We package this into an AAV5 and delivered by stereotactic intrastriatal injection in 6 ascending doses, as you can see here, denoted by cohorts A through F on the x-axis. And then 12 weeks following administration, we collected CNS tissues and assessed the striatum for AAV vector copy number, guide RNA expression levels, and leg B mRNA expression levels. And you can see we're getting a really nice dose-response curve as a result of that. And then we further assessed on-target editing at our exon 50 target site, again by looking at indels using next-generation sequencing. And we also assessed mutant Huntington's protein reduction by capillary electrophoresis. And you can see that we're getting a nice dose-dependent on-target editing as well as protein reduction, so about up to 50% indel rates on our target exon 50 SNP, and then up to about 80% mutant Huntington's protein reduction in the highest dose cohort. And then for delivery in the clinic, LifeEdit is working with Renishaw to employ their neuro-infused drug delivery system, which offers several advantages for acute intraparenchymal CNS delivery. First of all, the system utilizes convection-enhanced delivery, and it also has a reflux-inhibiting step, which means that the catheters can remain fixed in place for the entire duration of the infusion procedure. The catheter implantations and the infusions themselves can actually take place outside of the MRI, and all of the infusion lines can be run simultaneously, which really reduces the procedure time. And importantly to us is that the patients can be awake during infusions, and so we're reducing the overall patient exposure to anesthesia. And we also use this system in our 1-month tolerability and biodistribution study in adult cynomolgus macaques. And so just a note here that unfortunately the cynomolgus genome in the homologous HTT region does not contain the PAM-forming target T-SNP, so there is no editing to be assessed in these experiments. This is really a study focused on safety, tolerability, and biodistribution. And so we had 6 animals in this study, 1 that received vehicle, 2 that received low dose, and 3 that received high dose. And we're administering a total injection volume of 450 microliters per animal, and you can see between the caudate and putamen how those numbers break down. And so 4 weeks following administration, we collected CNS tissues again and assessed brain regions for AAV vector copies and transgene expression. In these plots, the x-axis denotes the tissue punch number, so several punches for some brain regions of interest, such as the targeted striatal subdivisions. We again saw a similar dose-dependent vector biodistribution and transgene expression in the targeted caudate and putamen, but we additionally saw a lot of expression in brain regions that are also vulnerable into HD, but which we did not directly administer to. We believe this to be retrograde transport of the AAV5 vector. And then finally, we also assessed the immunogenicity of our leg B nuclease protein, and so we did look at total antibody response in the serum and CSF following administration, and we saw no change in the antibody response. So in summary, we were able to package our system into a single AAV5 vector and deliver efficiently to the CNS in both mice and NHPs. In patient-derived cells, we observe allele-selective editing as well as reduction of the mutant protein, with wild-type protein levels remain unchanged, and we have not detected any bona fide off-target editing to date. In the transgenic mice, we observed really nice dose-dependent PK/PD, and we achieved what we believe to be a clinically relevant level of mutant Huntington's protein reduction. And we saw a similar biodistribution and transgene expression level pattern across vulnerable brain regions in the adult NHPs. And I also just mentioned that we did very recently meet with the MHRA and went over the LETI 101 program with them, which was very well received, including our off-target characterization strategy and our clinical trial protocol design, as well as our CMC strategy. And as we move this asset toward the clinic, we are actively looking for partners to help us move the asset forward. So if you're interested in learning a little bit more about the program, please feel free to reach out.
Excellent, thank you. Questions? Let's see, I'll start by asking, when you look in the monkeys, what percentage of medium spiny neurons actually have editing? Can you assess that?
Yeah, it's hard to say the exact number that have editing. We can say that we're covering like at least 80% of medium spiny neurons.
But I mean, how do you measure editing?
You'd have to do some single cell separation and then check each individual cell as opposed to a bulk lysate analysis.
Okay, questions? I think there's one here. Need a microphone?
So were you surprised that fibroblast edited around 20-30% and then the in vivo you saw cells 40-50%?
No, because we delivered with RNA transfection in those experiments because AAV5 has really poor transduction efficiency in vitro.
Okay. Maybe one other practical question. Because of your method of delivery, it's going to be difficult to do a sham control, I assume, right?
Yeah.
I'm guessing that's not part of the program, right?
No, it is actually, and we, like many of these companies, have made great use of the resources provided by HSG, and so we've worked, gotten a lot of scientific advice from Sam Frank and others about how we would actually employ the sham procedure. So it will be tricky to keep patients truly blinded, but it is definitely a critical part of our trial design.
Okay, that's good to hear. Any final questions? Okay, great. Thank you very much. Next is Joanna Osorio, Executive Medical Director from Sana Biotechnology.
Thank you.
Hi, everyone.
Here we are.
Hi, everyone. So I'm going to talk to you about the glia progenitor cell-based treatment for Huntington's disease. So changing gears a little bit for what you've heard so far. So my name is Joanna Osorio. I'm a neurologist. I have a PhD in glia biology. And this program is primarily based at the University of Rochester. So first, the lab started interested in Huntington's disease. The first question was to understand what is the role of glia in Huntington's disease? So we all talk about MSNs, and glia is always hidden in the background, but today we're going to give a little more emphasis. So we started attacking this question by obtaining two cell types, embryonic stem cells, from siblings. One, there was a healthy cell, Genea 19, that had normal number of CAG repeats. And then a Huntington's disease-affected embryonic stem cell line, Genea 20, which carries 48 CAG repeats. So what we did is to implement a model we use in the lab, which is to chimerize the mice with human cells. And so we use these cell lines, induce them to glia progenitor stage using a protocol that we have developed in the lab, and then transplanted these glia progenitor cells in the striatum of these mice, and allowed some time for these cells to populate the brain as they migrate and expand. And so what you see here on the bottom is human cells in as red dots, and you can see that they take over and they replace the mouse cells. So what we have at that time is a mouse that has humanized glia for the glia progenitor cells and also astrocytes that derive from them. So on the right, you have a panel, immunohistochemistry, just confirming that the human cells are there, and they do express markers of human glia progenitor cells, the proteoglycan NG2 marker, PDGF receptor alpha, and also astrocyte marker GFAP. So more interesting is to know what does these do, right? So we have the cells there, we humanize the mouse with human glia. And so interestingly, what we found, so these again were wild-type mouse transplanted with sick Huntington glia. What we realized is that these imparted a phenotype in the mice. So here what you see is results of rotarod, and the latency to fall was decreased in mice that were transplanted with the Huntington glia compared to all the other controls. So we wanted to understand why, what is really happening, and so we looked at the effect of glia on the host MSN. So these would be the mouse healthy MSNs that were then affected by the presence of Huntington glia. And what we saw is that there was evidence of hyperexcitability in the mouse host MSNs just by virtue of having human human Huntington glia in their vicinity. So we wanted also to understand why was this happening. And so in a parallel study, and this was purely done just on R62, we went to look at what is the connection and coverage of astrocytes within the synaptic cleft. And so for these, this study was done using electron microscopy, and we quantified the volumetric space of astrocytes surrounding the spine and synaptic clefts. And we found that there was significant changes, and they were actually significantly reduced in mature spines, not the same for immature spines, but there was definitely a change, which makes us postulate that astrocyte dysfunction could be on the basis of contributing to the MSN dysfunction. And so that led us to look at how do these astrocytes look in vivo on these chimeras. So human glia progenitors, some of them undergo differentiation to astrocytes. And what we saw on these Huntington-derived astrocyte cells is that they have much decreased expression of glia fibrillary acidic protein, so an astrocytic marker. And the cells that are positive for GFAP, they are aberrant, so they're less, and they're much less ramified, so suggesting that they have less ability to cover the synapses and to provide appropriate channel for transmission of potassium and other ions. So then we move to a more relevant question, which is, we know that glia impart some disease, but can we actually rescue some aspects of Huntington's disease if we would transplant healthy glial cells? And so for that, we went to a well-known Huntington disease model, the R62 mice, and this time we transplanted healthy human glial cells. And so we waited up to 20 weeks, and we showed by immunohistochemistry these cells have the phenotype we expect. And then, interestingly, what we saw is that we saw some significant results in the function of these animals, including motor phenotype by measuring in rotarod the latency to fall, which was then increased in mice that were treated with healthy glia. And there was also an increase in survival and the restoration of the striatum volume, which was decreased in the R62 model. And so we went back to what we have seen that glia can do, which is to hyperexcite MSNs. So we went to look at what does healthy glia do in the host MSNs of R62. Are we rescuing some of that? And so we did similar experiments with patch clamp, and then we found that the hyperexcitability of the host of R62 MSNs is partially restored by the presence of healthy glia. And because there was the suspicion based on RNA data that affected Huntington glia had a significant reduction in potassium channels, we looked at the interstitial potassium levels, and we found that while those were significantly increased in R62, upon transplantation of healthy glia, those interstitial potassium levels went significantly lower to the level of wild-type mice, suggesting that the healthy astrocytes and glia progenitor cells are able to appropriately channel the increased interstitial potassium and possibly through that effect reduce hyperexcitability of MSNs. So lastly, one of the most recent experiments in the lab, and also always with the intention of move this project translationally, was to think, okay, if we have human Huntington-affected glia and we transplant healthy glia, how are these cells going to behave? Are they going to outcompete or not? Because everything I showed so far is human in mice. And so what we've shown, what we've done initially was to tag 2 lines. So again, the G19 and G20, the paired sibling lines, one with normal repeats, one with higher repeats. And we tag one in green, one in red. And so initially we transplanted, these were wild-type animals, transplanted with Huntington-derived glia and allowed a long time for these glia to colonize the brain. So all the brain was green, green meaning Huntington-derived glia. And then after 36 weeks, we transplanted these same mice, so retransplanting these mice in the adult age with the red cells that are wild-type glia. And then we studied over up to 72 weeks the effects of this competition and replacement. And so what we saw, and you can see here in the images on the bottom, is that progressively over time there is takeover of these red cells, so the wild-type cells, over the previously engrafted Huntington-derived glia. And what we saw is that this is really interesting. It goes as a competition such that the healthy cells go through a line, so it's almost just a very delineate border, and then at that border we see some signs through tunnel staining of apoptosis in the cells, the Huntington cells that are at that border. So there seems to be a competition effect that the healthy cells are promoting apoptosis of the Huntington cells and then continuing to replace those. And so this was very encouraging for us to think about a translational approach, which is where we are at this point. And so for that, we had the protocol previously published to generate human glia progenitor cells out of pluripotent stem cell sources. And now we have that protocol GMP compliant. And this protocol basically follows the different stages in development, from neural progenitors to glia early progenitors and more mature glia progenitor stage. So at this point, our goal is to advance to a clinical trial, for a first-in-human phase 1 trial. We're not there yet, so we're still waiting for the support to start. So in terms of clinical trial readiness, we have some efficacy studies, some of those I showed you, but we're working on some others, definite efficacy studies. And also we have an ongoing safety tox study as well as delivery study, as these cells would need to be delivered via neurosurgery intracerebrally. And so our plans so far, which are not completely final, but we intend to do a phase 1 open label dose escalation study targeting patients with HD-ISS stage 2 to mild 3. The intervention would be the transplantation of these GMP-compliant human glia progenitor cells along with immunosuppression, probably for a period of 12 months. And this is because the cells is an allograft, and so we would need to prevent rejection, at least temporarily. And the outcome measures proposed, of course, primary safety, but then as key secondaries, we're looking at key measures in the field, the composite UHDRS, TFC, HD-CAB, and quality of life, as well as other, including imaging and some exploratory CSF biomarkers. And so in summary, I showed you today that human HTT glia leads to MSN hyperexcitability and motor impairment in otherwise healthy chimeric mice. I've showed you that healthy human glia can improve the motor phenotype and hyperexcitability of HD mouse models, in this case the R62 mouse model. And lastly, that healthy glia can outcompete previously established HD glia in chimeric mouse models, suggesting a therapeutic strategy for replacement of disease glia in Huntington's disease. So, oh, sorry, there was one last slide. It was just the support. Okay, it's gone. But so thank you for the support from Sana Biotechnology, CHDI, the Adelson Foundation, and Novo Nordisk, and everyone in the lab who has highly contributed to all these projects, which their name was in the slide. So, and from Steve Goldman's lab. Thank you so much.
Time for one quick question.
I'll be around if questions come later.
Don't see any hands. Well, here's a question for you. How can you control the possibility that the transplanted glia might replace more of the cells than you want and might actually disrupt the cell populations in the striatum?
Yeah, that's a very good question. So Everything we've seen so far in the lab, we have done studies modeling Huntington as well as myelin deficient diseases. And so these cells seem to be biologically very smart, that they act so they behave in a phenotype that relates to the area where they are. So the cells that are in the striatum, it affects the fate of these cells and what they become. So it affects their differentiation and affects their migration patterns. For example, when we transplant in the corpus callosum for myelin diseases, we see these cells migrate throughout, and once they fill out the corpus callosum and white matter, they kind of stop. Their mitotic index decreases over time, and the same for striatum. Once they fill the striatum, their mitotic index by then is slowed down and decreases, and so that we don't really see invasive tumor-like effect of these cells.
Thank you.
Thank you.
Next to Juha Savola, VP of Clinical Development for Neuroscience and Ophthalmology at Spark Therapeutics.
Chairman, dear friends, colleagues, this is a great day for me. Years of hard work in the team to come here and introduce you how Spark Therapeutics is stepping into treatment of neurological disease, especially Huntington's disease. Spark Therapeutics is best known for introducing the first gene therapy for treatment of hereditary vision loss, 2017, and now we are opening the door in treating another so far untargetable disease, Huntington's disease. Today I'm updating you where we are with SPK 10001 on its way to first-in-human study. Bottom line of the disclaimer is that nothing in this presentation on SPK 10001 shouldn't implicate that we know anything about its efficacy or safety. I'm employed by Spark Therapeutics, and I'm not going to walk you through the pathway of Huntington's disease, respecting the share's ask, but I'm making a point that scientific evidence is strong enough to make a hypothesis that mutant huntingtin protein is a key player in the part of the toxic pathway resulting in neurodegeneration, death, and dysfunction of striatal cells, resulting in clinical manifestations of this disabling disease. And we predict that by reducing the amount of that mutant huntingtin protein, we can provide benefit for the patient. And that is what we are seriously targeting in our clinical program. SPK 10001 is AAV-based gene therapy, and it is designed to form microRNA in those cells where it is getting into, in caudate and putamen. And microRNA targets the messenger RNA coming from the expanded huntingtin gene. By that formation, that messenger RNA is not read and no protein is formed. Preclinical evidence is showing, as highlighted in this slide, is showing that when we administer SPK 10001 into the caudate and putamen, we see nice dose proportional increase in the genome of SPK 10001 or microRNA, and proportional reduction in huntingtin messenger RNA and huntingtin protein levels. These data are from non-human primates. When we look these data at 3 months, at 12 months, we see that any of these readouts are very comparable at these 2 time points, meaning 3 months is already fast or early enough to demonstrate stable pharmacological action, and the effect remains so-called durable for the first year at least. A note on the technical approach. SPK 10001 in these non-human primate studies had been administered using a neurosurgical approach where we position ClearPoint smart flow cannula along the longitudinal axis of the caudate and putamen. There's one cannula to caudate, one cannula for putamen on both sides of the brain. 4 cannulas, as you can all calculate. Once we reach with the cannula on both sides of, say, caudate, we start infusing SPK 1001, and with a few steps, we infuse as we go to the other end of the target. And in monkeys, we have seen up to 80% coverage in these targets, coverage being measured by caroterial imaging, MRI imaging agent. Bottom right, you can see a couple of histology pictures, and you can see there's supposed to be the track of the cannula there, but you don't really see it. The brain structures tolerate this really well, this approach we have been using. Another important finding for me has been to see how robust, how predictable the biodistribution is when using this posterior temporal approach to infuse SPK 10001 into these structures. Here on the right side, you see individual monkey brain slices, and you can see low dose, mid dose, high dose. Individual animals, and those look at the red dots, for example. I find them easiest to find. And you find that the intensity of those red dots between the animals is very much the same, meaning that, yes, we do get the same response in each of these animals. It's also interesting to learn of the behavior of this microRNA and transgene expression biodistribution in different brain regions outside where we have infused that. And this is the part of the biology of these capsids or transgenes. There is anterograde, maybe retrograde transportation. We put them into anatomically well-defined positions, as I described. Good coverage there. We don't spill over, but the globus pallidus, frontal cortex, thalamus, all those are getting the signal. They get the transgene, they reduce messenger RNA, they reduce Huntingtin protein levels in these structures. This could be meaningful in the context of Huntington's disease pathology, and to have this distribution outside the primary site of infusion.
2 minutes.
Thank you. And last, we haven't seen any increase in the lateral ventricle sizes across the 3 doses we have tested in preclinical models. Now, browsing quickly through some clinically important features of the program. In this program, we are addressing the well-known wide distribution in the individual distribution of the caudate and putamen volumes by looking how many vector genomes we should be administer per cubic millimeter of the brain structure. We evaluated the monkey data we just have been collecting, expressing the vector genome per brain as vector genome per cubic millimeter of the structure, come up with a statistical model to estimate the dose response curve. And using this, we are selecting the doses for clinical program, low dose representing 25% mutant huntingtin reduction, and high dose 50% huntingtin protein reduction.
1 minute.
Thank you. Time flies here. Patient selection criteria, couple of points. Inclusion criteria, CAC repeat length, as no surprise. Stage 1 criteria, based on HTISS criteria have to be met. TMS, TFC, representing a typical early manifest patient population. Concomitant medications allowed. We exclude patients where we are not confident that we can safely administer or put the cannula because of anatomical features. Past antisense oligonucleotide accepted if that is over a year ago. Study is in 4 segments. Part A, open label, low dose, high dose group. High dose continues in the randomized placebo surgery control. Part B, those control subjects will continue in the crossover delayed start design into the active treatment in Part C. Everybody who has received SPK 10001 will be followed in a long-term follow-up study. Neurosurgery is used, intraparenchymal administration based on smart flow cannula and the navigation system of ClearPoint. Study outcomes, TFC, looking for our preliminary efficacy. We are checking Huntington's disease digital motor score, if that could be an appropriate endpoint to detect a signal of efficacy, and using composite UHDRS also for that purpose. Most importantly, I want to thank this community of Huntington's disease, scientists, patients who have helped us to understand key features for designing this program. Especially I want to thank our colleagues in the Roche team, Peter, David, and Jonas, people outside the company. We have received extremely supportive feedback from various experts in the field. And thank you, and I'm excited for the opportunities to come during the coming years to update where we are with the program.
Okay, great. Sorry, but we're out of time, though, so no time for questions. One last talk. Don't want to run too much over. We're already a little bit behind. Next is Serena Hung, Chief Medical Officer at Atlanta Therapeutics.
Good afternoon. My name is Serena Hung. I'm the Chief Medical Officer of Atlanta Therapeutics, and I want to thank HSG for giving me the opportunity to tell you a little bit about what Atlanta is doing. So the disclosure is that I'm a full-time employee of Atlanta. So just to tell you a little bit about Atlanta, we are a biotechnology company located in the Boston area. We are dedicated to using our unique di-siRNA oligonucleotide platform to target central nervous system diseases. The current areas of focus that we're working on is, number 1, Huntington's disease. We're also working on refractory severe pain and also a genetic developmental epileptic encephalopathy. I always trip up on that one. So, as you probably know very well by now, that we're using a small interfering RNA technology. So, as you've heard before, RNAi is a natural cellular pathway to knock down genes and protein expression. And RNAi uses synthetic duplex siRNA molecules to degrade target mRNA. And they're synthesized with an active antisense sequence complementary to the target transcript or transcript of interest and a carrier sense strand. And on the right-hand side is a diagram to sort of outline, how it works in a cartoon form. So you can see the siRNA, and then it finds the RISC complex, and basically help the RISC to find the target transcript, and it leads to reduce degradation. So historically, with oligonucleotides, there are certain delivery challenges. Number 1 is intrinsically, there's poor distribution and very rapid clearance time, so it makes it difficult to get to the target tissues. Also, another thing about oligonucleotides is that antisense oligonucleotides do not distribute well beyond the superficial regions of the brain. We've licensed in the chemistry based on the technology at the University of Massachusetts Medical School. In fact, our scientific co-founders are Craig Mello, who won a Nobel Prize, and also a couple people who, in this audience you might know very well, Anastasia Korova and Neil Aronin. And we spent a few years optimizing the chemistry such that we can improve the CNS distribution, so it can distribute very well into the spinal cord and as well as the deep brain structures. We also managed to improve potency, durability, and tolerability, and I'll show you some data. We also use ionic conditioning formulation to expand the therapeutic index. Another thing is that we don't need to use conjugates or LNPs for delivery, which makes manufacturing a lot easier. I don't need to tell you about the rationale why we're using siRNA for targeting HTT, but I think one of the important things is that with the excellent distribution, we have the ability to knock down HTT in the deep brain regions, including the caudate and putamen. Also, we have excellent durability, which makes a lot of sense, especially since we're delivering it through intrathecal delivery. We want the dosing frequency to be as convenient as possible. So we're looking at 6 months or maybe, even less frequently. So this is one of the experiments where we looked at delivery of a couple of different dosing regimen in non-human primates. So we have the study design on the left-hand side. We have the control arm, and we have one arm that received 50 milligrams of ATL-101, and we take them down at various time points so that we can look at the course of change in HTT. So we take them down at 1 month, 3 months, 4 months, and 6 months. We also want to look at repeat dosing, not only to look at the effect, but also want to start evaluating repeat dosing safety. So we administer 3 doses of 50 milligrams of ATL-101 and take them down 1 month after the last dose. So as we were talking about the deep brain region, so I want to show you the deep brain region first. So on the blue bars, we'll start with the blue bars. Those are the monkeys that received the single dose and then take down at various time points. So you can see that the maximum lowering of HTT happens at 4 months. At around 6 months, there is actually still about a 50% reduction. But at the 4-month mark, the reduction is about 75%. Now, the purple bar are the monkeys that received the multiple doses, 3 doses, dosed 3 months apart. And you can see that in the caudate and putamen, the lowering is actually also at around 75%. So in the cortex, we have even more impressive knockdown. So the time course is similar to what you see in the deep brain. So, but the knockdown, the maximum knockdown in this case is around 80 to 90% with the single dose and the multiple dose. Of note, there was no safety signal coming out from any of these monkeys. And in our other experiments, which I'm not showing you here, we've dosed even higher doses, and we still haven't seen any safety events in these monkeys. One thing that we also wanted to look at is inflammation. So we looked at NFL levels in the CSF, and these were done in the monkeys that got the single dose, and we inserted a catheter so that we can draw CSF every month. And so you can see that NFL levels did not rise throughout the whole 5-month period. The last time point is day 150. This is despite the fact that they had a 75 to 90% HTT knockdown. We've also looked at RNA-seq and specifically looking for inflammatory signatures, and we also didn't see any abnormality. We've done some modeling just to start thinking about what kind of clinical doses we're going to need. We use a 3-compartment model scaled by CSF volume, and currently with this model, a 50 milligram clinical dose will knock down HTT by about 50% for 6 months. We're continuing to refine the model using our ongoing data generated from our ongoing experiments. So in summary, the non-clinical studies support development of ATL-101 as a disease-modifying therapy in Huntington's disease. A single dose of ATL-101 di-siRNA delivered intrathecally in NHPs produced greater than 80% HTT knockdown in the cortex and about 75% in the striatum. And the knockdown was maintained for over 6-month period for about, down to about 50%. There was no safety signals observed, and NFL did not increase with HTT knockdown. In modeling the clinical dose, a 50 milligram dose, single dose, can lead to about a 50% knockdown for a 6-month duration. We plan to initiate clinical trials in HD patients in 2025, and we look forward to working with many of you in the coming years. Thank you.
Great, thank you very much. We have time for a couple of questions. Let's see, right there. Why don't you get up and yell? Well, take the mic. I don't think we heard you.
I couldn't hear you.
Let's talk about storage and delivery of these RNAis for patients, because they're not that stable.
Actually, currently, based on our current work in CMC, they're actually pretty stable. Of course, we're still doing a lot of work to make sure that they indeed maintain the stability. But we anticipate being able to deliver these drugs, to the clinic in vials, basically.
Okay, time for one more question over here.
Hi, Serena. A beautiful talk and really impressive data. I didn't get, and maybe you can't tell us, where your siRNA targets in the Huntington.
Oh, if you want to know whether it's exon 1, no, it does not.
That?
No, we're not concerned about that, because I think based on a lot of data that has been generated through the years, knocking down mutant Huntington would have a significant effect. And currently, there is some ongoing exploration with the exon 1 hypothesis, but I think just knocking down mutant Huntington to a sufficient degree should have a beneficial effect.
And it's total Huntington lowering as well, correct?
Yes, it's total. Yeah.
Great, thanks very much. Thanks to all the speakers, to the audience, and to Sarah for the nice introduction.
And thank you, Dr. Ross. All righty, up next, we are happy to have a new session this year that will highlight some of our poster presenters. Following these presentations, we invite you to stay for a special evening of networking and reception for all of our guests, which is you. Sorry, Dr. Ross, we don't have a DJ this year, so we can't see your funky moves. More on that after this session. Day 2 kicks off tomorrow morning promptly at 8:30 a.m. with a welcome from me and then Dr. Stephen Pantalot, who will lead our first session on contemporary clinical challenges in HD. Breakfast foods will be available in the main foyer beginning at 7:30 a.m. And now, please welcome back our CEO, Dr. Daniel Clausen, to introduce our poster presenters.
So we have 3 presentations of work ongoing with CHDI, and I think you'll find them very interesting in terms of the breadth of the work, especially as the Enroll 2.0 is getting ready to come out. But the first presentation is an update in PET imaging with Robert Doot. Thank you.
Hello, everyone. Today I'm going to give a brief update on some of the PET imaging going on in Huntington's disease. I have no disclosures. PET biomarkers, they offer a window into the progression of the molecular pathology. And there's 3 different targeting areas that I'm going to talk about today. First of all, targeting aggregated mutant huntingtin. The reason why we're interested in this is because it's causal in the disease pathway, and also allows us to do a direct readout of the mutant huntingtin level and any response to therapy for things targeting these areas, as it's an area of great interest. However, it's challenging due to the target density. Secondly, you're going to talk about a particular tracer that's looking very interesting for biomarkers of striatum, the phosphodiesterase 10A. That's the most developed, and it shows some early changes. And then finally, I'll talk about some more areas that target more of the cortex, and we can look at some global effects, and talk about some of the work that we have ongoing with some CHDI sponsor studies to identify which are going to be useful potentially for clinical utility. So when you're starting with new bio tracers, in this case PET, you have to go through preclinical, first in human, and this is only if at each step, and each step gets more difficult to show that it's worthy of going on. In this case, we're interested in mutant huntingtin, the aggregate forms. We've had enough on the biology today. So one thing I want to point out for this target, what makes it especially difficult, is its low abundance. So for perspective, the amyloid beta aggregates that are of interest in Alzheimer's have like 100 times higher concentration. Doesn't make it impossible, it just makes it more challenging. And here's where we are. We have the first generation 180, and which we've just recently finished that human study, and I'll talk about 3 of our next second generation tracers about to go into humans next year. So before you go into humans, it's very important that you demonstrate success in animal models. And on top, you'll see success in mouse. It's clearly a difference between the mouse model on the left and the control on the right. Similar with the macaque and non-human primate in the control. 180 over in Leuven in Belgium, we went through up to the 3rd no-go, where following those evaluation, the quantified differences between the healthy controls and the participants with HD was just not sufficient to continue the study. But fortunately, we're ready with our second generation radioligands. All these second generation radioligands have improved binding to aggregated mutant huntingtin and less off-target binding. And most of them have replaced the C11 PET label with a much more clinically friendly F18. And if you want to know some of the specific chemistry, the ways we got from first generation to second generation, I encourage you to look at Liu et al. In 2023, and they'll walk you through the specific ways we got from the first generation to second generation. So starting next year, we'll do one of our first second generation 009. It's a C11 PET tracer at Leuven. And in parallel, in America, at Baltimore, we'll be looking at 2 F18 radio tracers, 650 and 385, in HDISS stages 2 and 3. So any of you interested in these studies, reach out to myself or Chris Ross and G. Bang, who are here today, if you want more information. So on to the second area of targeting, the striatum. I'll walk you a little bit through this study because it's a very compelling preclinical study. So if you look in the middle, you can see 2 hemispheres of the mouse model. On the left, you see one that had a surgical procedure to inject the treatment, and on the right side, it had a similar procedure to do the control, and you see that there's a higher uptake of the treatment. That's what you're going to see on the left when you see it in the more yellow-colored range, that with this treatment that was supposed to lower the mutant huntingtin, we saw a reduction of about 40%. And here's where the PD10A is showing its promise as being perhaps a more general tracer for evaluating treatment success. It had a similar magnitude of about 40% increase. And if you look to the right, here's where you intervened in the mouse later at 5 months versus 2 months, you saw a similar pattern. When you had the reduction in mutant huntingtin of like 25%, When you went with PD10, you saw a similar magnitude of positive 26. This makes us really encouraged that this could be a good general tracer for especially things targeting areas in the striatum. Another area of interest is some of the areas in cortex. When you're looking at the cortex, you're probably looking at a more global effect. We have done some studies with UCBJ, a C11 tracer, at Leuven, and this next upcoming study is just starting this fall. Eye synapse, in addition to some MRI properties we're comparing, we're interested in looking at the C11 tracer versus an F18 tracer with the hopes that the F18 tracer will be close or better, but just close will be enough because F18 tracers are just much more clinically friendly. IMARC. This is another ambitious study that we have ongoing at King's College London. What's really great about this study is we're going to look at 4 tracers of interest in people with Huntington's disease as well with healthy controls. So you're going to see all 4 tracers, and you're going to see them at baseline, plus longitudinally 1 year later and a second year. Another really great aspect for this is if we continue to see the positive results, we're going to look at much earlier in the progression of the Huntington's disease patients, looking at stages from 3 all the way up into earlier 2, 1, and 0. So this is a very exciting study, and 3 of them are more or less looking at cortex. There's a serotonin 5-HT2A receptor, another one histamine H2R, a cannabinoid CB1, and then as already foreshadowed, the PD10A is of interest. We're going to look at PDA10, which is one more for the striatum. So in summary, the molecular biomarkers of striatum, that PD10A is looking very positive. However, we're doing additional studies now to nail down what is the best quantification and start to estimate effect sizes if you want to take these into larger clinical trials. For biomarkers of cortex, the SV2A, the serotonin, the histamine one, the H3R, and the cannabinoid 1 receptor are all also looking positive, and we continue to evaluate them. And then for measuring mutant Huntington's, the first generation got us part of the way, and now we're very excited to take the second generation into studies in humans in parallel next year. And of course, this didn't take a village. It took many countries to get this far. So thanks, everyone.
Have time for some questions? Anyone have questions? You're right, Chris. It's really blinding.
So I have a question about how you envision in once you have your second generation Huntington aggregation ligand optimized, let's call it, in 2 or 3 years. What would you see its role being in both the early development and then in the clinical trials of all these therapies that we've heard about today in terms of potentially accelerating their development and bringing it earlier into the clinic, into ISS 0 stage patients? Can you talk a little bit about that?
Sure. So as you notice, we're not just looking at one tracer. We're looking at a couple of them, and some are perhaps more suited, at least at the preclinical level, at answering different questions. So part of what we're doing these first in humans, we're validating them for humans, and we're going to try to choose if hopefully there's just one ring, one compound to rule them all. But if not, we will identify which particular tracers are best for specific questions. So depending on where you're looking at longitudinal measurements or other areas. So that's a key part. What are the best tracers for different compartments? Then it might get down to what are your specific patient populations of interest. So some tracers might be better at, say, the C11 tracer. Say that absolutely has the highest sensitivity to change, and you have something you think it has a low effect size. Well, we're going to have a toolbox, and we're going to say maybe use a C11 with the constraint of there's not every site that's going to be able to do that imaging. As we continue to go out to larger and larger phase studies, then you're going to hopefully be able to transition, if it's not already the winner, to some of these F18 label tracers. And that's because that change in half-life from 20 minutes to 2 hours makes a huge difference. It means instead of only having patients being able to be imaged at large academic sites, you'll be able to have like a regional center, like say you want to do it in Montana, you could have University of Utah Cyclotron make you that compound and ship it out, but you couldn't do it as the C11. So if it turns out the C11 tracer is the best one, part of our work may be, can we relabel it? So it's all about getting a toolbox and you telling us what you need, and we'll work on the next tool.
Thank you very much, Robert. Now we get to hear about Enroll 2.0, and Celine is going to take us through that.
Okay. Well, thanks HSG for giving us the opportunity to present on these very exciting changes to the Enroll HD protocol. So, as many of you know, Enroll HD is a global observational study as well as a research platform designed to collect data and sample that are then shared with the wider research community. The objectives of Enroll HD are, in fact, to improve the understanding of the mechanism underlying HD. The second objective is to develop guidelines that will then inform clinical decision, improve clinical care, and the impact that the disease has on the family. And the third very important objective is to provide support for the design and the conduct of clinical trials. Now, I want to highlight that the objectives in the new protocol haven't changed. They're very, very much the same. So what has changed is, and why we have implemented this, the amendment is, you have heard about all these new trials coming up and all the programs. So all of this, most of these trials will focus on participants in early stages of the disease. So we want to make sure that Enroll will have the right cohort distribution that will enable to meet those needs. So I cannot stress enough that it's not a new protocol, it's a protocol amendment. So it's the same study with some, I would say, significant changes, but it's the very same protocol. So what will change, so the participants will continue to come on an annual basis. What will change is that the protocol will include the participants will be included in different cohorts, and each cohort will have a targeted battery of assessment and procedures. Some of the visits may be remote, depending on the cohort to which the participant is assigned. We have also introduced a different collection of targeted biosamples, as well as sub-studies, which I'm going to talk in a minute. So these are the three cohorts. Cohort age will include participants in HDISS 0 and 1, and participants that haven't been tested, so at risk of HD. And cohort B will include participants in HDISS 2 and early stage 3. And cohort C will include participants in late stage 3. And then we have controls that based on the age, whether they are younger than 40 or older than 40, will be assigned to cohort A and B, and they will follow the respective schedule. And then for cohort C, the companion will follow, the companion to the cohort C will follow the cohort C schedule. Cohort assignment may change during the course of a participation in the study based on the progress of their disease. So what we are trying to target is to bring the current cohort to a composition that will envisage a higher percentage of participants in the early stages. So we would like to bring the participants in stages 0 to 2 from, currently we have a 28%. We would like to increase it to a target 51%, whereas we would like to reduce the HDI says on stage 3 from 48 to 34, as well as the control from 24% to 15%. So I'm not going to go through all the evaluation and assessments, but as you can see, they are targeted for each cohort. Cohort A has a reduced battery. I forgot to mention that cohort A participants will also be offered on alternative years remote visits. PROs, in order to optimize the time at the site, the PROs will be, except for the baseline visit, PROs will be completed by the participants at home. So we expect the cohort age visit to take about 45 minutes on site, plus the PROs that are completed at home. The cohort B is the cohort that will go the more extensive assessment. We envisage that the visit on site will take about 1.5 hours, again, with the PROs at home. And cohort C, again, except for the baseline visit, will be offered the remote visits, and it will take about 45 minutes. For cohort C, we will use this HTCF and the HTCSQ, which my colleague Kieran is going to talk about in the next presentation. So as I mentioned, we have introduced targeted biosamples. So so we have two, similar to what we have in the current protocol. All the participants will undergo blood collection for genetic research genotyping. If they have done it for the current protocol and they're just transitioning to 2.0, they won't need to redo it again. If they're recruited de novo, they will have the blood 6 meals collected for research genotyping. We have introduced a slight change. So we've introduced CAG on saliva, which would be done in exceptional cases where the blood CAG, the blood collection cannot be done. So in addition to the core biosample, core research genotyping that is done on all the participants, we also have included targeted biosamples that are optional. They're conducted at selected sites for a given period of time on a targeted subpopulation. In any event, the total blood collection will not exceed 50 mils. And you can see in the table, so we have, as a target biosample, blood collection for whole blood, RNA, and plasma. So we will continue the collection that we are currently conducting under the current protocol. The new protocol will envisage saliva, tears, urine, and feces. As I mentioned, the new protocol also envisages 3 sub-studies, which are nested within the protocol. So the actual protocol includes the sub-studies protocol as an appendix to the main protocol. As a nested study, they basically use the same, the infrastructure and basically the piggyback of the role, basically infrastructure and the protocol. They will be offered at selected sites and conducted only on some participants. So the 3 sub-studies are Origin HD, which is a semen collection and blood collection from male participants over 3 consecutive visits. And then we have the iron roll, which is the imaging sub-study that will collect MRIs from over 5 consecutive visits. And then we have pedigree, which is the family history that is only completed once, but may also be updated in the future, in future visits. Depending on the cohort that the participants are assigned, the sub-study may or may not be offered, so the Origin HD and pedigree will not be offered to the control, and the iron roll sub-study will not be offered to the cohort C. And as you can imagine, it's a major undertaking, so it really takes a village. So I would like to acknowledge the work of Christina Sampaio and her team, as well as the imaging team, the fluid biomarkers, the clinical outcome, the operational team, and I'm sure I forgot someone, but it really is like a collective big team effort within CHDI.
take one quick question for this one. Unless it's good. I think you answered all their questions.
All right.
Thank you for that. That was great.
Good.
Thank you.
Thanks, Annie.
Great, and the last presentation will be late stage assessments. Come on up. Yeah. I think it's ready.
Okay, great. Thank you. All right. Hi, everyone. My name is Kieran Borker. I am a clinical program manager at CHDI, and I'm here to talk about the later stage assessment study, or the LSA study. I don't have any financial or non-financial disclosures. So as we all know, HD is a neurodegenerative disorder that impacts an individual's motor and cognitive abilities. And as HD progresses, an individual can become increasingly dependent on their caregiver, whether informal or formal. And when we look at this population within the Enroll-HD sample and look at the data that is provided, we notice that this population is taking a larger and larger percentage of the Enroll-HD data, and some of the assessments are not tailored towards these individuals. So the motor or verbal assessments are quite cumbersome or arduous, and that can lead to some missing data points and potentially not asking the right questions to this population altogether. So because of this missing data and information, clinicians and HD researchers are not able to fully study how HD progresses within these later stage individuals. And while caregivers do have a wealth of information about the clinical and behavioral characteristics of HD individuals, there's no currently validated assessments that leverage this feedback to measure the change in HD patients in stage ISS stage 3. And that has basically led to the development of two assessments, the HD-SIF, which is a tool that encompasses the 3 UHDRS functional scales, the functional capacity, the functional assessment, and the independence scale. And this is an assessment that can be administered either remotely or in person to the caregivers. And the second assessment is the HD-CSQ, or the Clinical Status Questionnaire, which is essentially an interview between the companion and a certified rater to capture some essential information on the clinical status of the individual with HD. So the objective of LSA is primarily to evaluate the internal consistency, reliability, and validity of these two assessments. And if this objective is reached, then we can say that we have created assessments that measure disease milestones and progressions in people with HD ISS stage 3. And we can include these assessments in larger scale observational studies or clinical research. And we can also determine whether companion-reported assessments are valid and can be administered remotely as well as in person, allowing for flexibility within the companion participant. So the LSA study itself is broken up into two parts. The first part, the goal is to evaluate the internal consistency, reliability, and validity of the HD-SIF assessment using a crossover design. So we, during the baseline visit, have the HD-SIF assessment administered by Rater A, and Rater B administers the UHDRS functional scales. Each rater is blinded to each other's scoring, and we also have randomization and group administration numbers. And then during the remote visit, that's a follow-up visit where the HD-SIF assessment is administered through an external third rater. And there's a total of 20 dyads, which essentially is the HD participant and the companion participant in part 1. Part 2 assesses the clinometric properties of the HD-CSQ and the HD-CIF using a cross-sectional design. We are currently recruiting for Part 2 with the goal of 150 dyads. The baseline visit will be the same day as the HD participants enroll visit, and only the ICF is basically just consent is collected. And then during a follow-up visit, which is administered remotely, the HD-CIF and the HD-CSQ will be administered for the companion participant only. And in terms of inclusion criteria, the HD participant needs to be an Enroll HD. They have to have a DCL score of 4, a CAG repeat length of at least 36, and an inclusion score of 16. And the inclusion score is basically derived from the functional assessment scale and the total motor score. It ranges from 0 to 50. And the companion participant has to be at least 18 years old and have sufficient interaction and knowledge of the HD participant. So as I mentioned, we did recently complete LSA Part 1, where we successfully recruited 20 dyads. 21 dyads were screened, and 20 successfully completed the assessments because one dyad did drop out or was lost to follow-up. And this was recruited at 4 clinical sites in the US. For Part 2, we are still recruiting. We currently have 42 dyads as of the 1st of this month across 6 clinical study sites, and we've also activated a handful of sites in the UK. So we currently have 7 activated and recruiting sites, with another 11 sites in the US and in the UK in startup. And quite recently, we met with all of these sites who are activated and is currently starting up the LSA study during the EHDN Enroll Congress in Strasbourg. And we really got to understand what are some of the recruitment challenges and the strategies that they implement at their site when you're working with HD participants in later stages. And I've just summarized a few of the key takeaways from that meeting on the slide here. But the main and most prominent challenge that was reported was that the LSA recruitment strategy does conflict with the current Enroll HD strategy focusing on younger pre-manifest at-risk individuals. So that is something that is an ongoing discussion between CHDI and the clinical study sites, and we always encourage our sites to provide feedback to us so that we can support them in any way possible. So I'd just like to mention that I'm part of a larger team here that couldn't fully be represented. I help manage the US sites, but we have 2 study managers in the UK and also our lead project manager. And finally, I just wanted to say thank you to CHDI, Dr. Frank, the LSA study team, the protocol advisory team, and of course our clinical study sites and all the HD participants and families that make the LSA study possible. So thank you.
Good.